Hello, everyone, and welcome to another lecture for Envs2001, our Lab and Field Methods course. This week, we're going to be moving from the aquatic environment into the terrestrial environment, and we're going to change our focus from water to soil. Now, this is not exactly my area of expertise, so this lecture will be a bit shorter. However, I think it's important for us to study this topic because it affects us in our daily lives and it is critically important for environmental science and the study of our biosphere. Now, why soil? Well, we should all know the answer to this. Soil is really the underpinning of our food security. Most of our crops are grown in soils, and so we rely on them very heavily for food and for maintaining our human population densities. Soils are also important because they are useful in biogeochemical cycling of certain elements and compounds, and some of those being pollutants. The soils can help us to mitigate pollution by hosting organisms that can break down pollutants or by directly absorbing them, which we'll talk about later. Soils provide a natural means for filtering water. So precipitation and even effluents from sewage and industrial applications can be really cleaned to high purity by simply moving through soils and moving through groundwater. The earth itself can be a very effective filter for water. Soils are also important for carbon storage. There's a tremendous amount of carbon locked away in the soils of our planet, and keeping it there is something that is a big concern to climate scientists because as the earth warms, the decomposition of soils will accelerate. We'll talk about that as well later in this talk. Last but not least, soils are non-renewable. They're non-renewable resources. So you will often hear people talking about soil conservation, preserving what soils we have, protecting their quality or improving their quality over time so that they will be there to be used for all these ecosystem services in future generations. Now, I just used the term ecosystem, and that's because soils are a key factor in ecosystem productivity. Soils reflect the type of bedrock that underlies them, the bedrock type and its composition. And if you're interested in geology, you might be more familiar with certain rock types, the bedrock types that underlie soils in this region and other parts of the world. Soils can reflect materials that have been transported from elsewhere. So if we think about geological time, soils can be pushed around or derived from other processes. For example, glaciation. When glaciers form over large tracts of land and then retreat, that friction, the force between the ice and the landscape can create soils and can transport soils across vast distances, which can make the landscape more heterogeneous. These processes affect soil texture and its chemistry and the rate at which nutrients can be supplied to the plants. So plants and their root system, vascular plants, and their root systems are taking advantage of nutrients that are locked within or adhering to soils. So all of this variability often drives variation in ecosystem processes. So if we want to talk about the productivity, the primary productivity, the amount of plant biomass, the plant community composition, the competition between plant species, and the successional process by which terrestrial ecosystems change to hosting different plants that are dominant within that ecosystem. And as plants are the basis for the terrestrial food web, then we can see really dramatic changes in the types of consumers and predators that we'd find in ecosystems. And all of it's being derived from the productivity, which is a key component of, or a key service that is provided by soils. Now, when we think about environmental science or environmental soil science, there are a variety of different terms that come to mind. So here we have listed water quality. Soils are a key component of maintaining water quality either in groundwater or in tributaries, in rivers, streams, even in the ocean. Soils play a role in wastewater management, in stormwater management. They can provide buffering capacity or filtration capacity for these types of pollutants. When we think about soil science, we have to be concerned about erosion control. We want to retain soils, not lose them. We have to be concerned about metals, heavy metals, and pesticide contamination, which can bind to soils and be persistent for a very long time. Soils science is related to wetland restoration, which is a key area that provides the interface between the land and the sea. Soil degradation, or a lowering of the quality of the soil, is a big concern. Nutrient management, how much nutrients do we need to retain in soils? How much do we need to supplement in order to use soils for agriculture? And what's happening within the soil itself, the ecosystems that are inside the soil, the microbial ecology, the fungal ecology, all of these organisms play a key role in the cycling of important nutrients in particular, and the decomposition of organic matter. Can we use soils to improve the situation? Can we use soils to clean up or bioremediate large or chronic pollutants? Can we genetically engineer plants that can help in remediating polluted soils? Can they remove contaminants and therefore allow us to extract important pollutants from soils? Land use change, global warming, acid rain, all of these things have in some way a relationship or a key impact on soils. So this is why we're going to spend a week studying soils. Now, if we were to take a slice of the earth, the soil would be a very, very tiny layer at the very surface. And if we can imagine a soil profile, we can see here in this image there's the green grass is growing on the surface of the planet. This is the area that we're most commonly interacting with. And then below the surface, we have a whole gradient of change in the soil profile. And typically, we are moving from a very organic-rich surface layer into a transitional zone that is comprised of increasing proportion of mineral soils to the point at which we reach the bedrock, which is exclusively mineral and in the complete absence of organic matter. So let's go through this in some detail, and I think this will be informative for you if you're interested in soils, but primarily today we're going to be working with the organic stuff and the surface soils at the very top. So that first layer at the surface is called the O horizon, and this stands for the organic horizon of soils. And the organic horizon is comprised of perhaps a layer of litter, which could be from leaf litter or grasses that have died and slowly been packed down to the surface. So these are non-decomposed organic matter that can accumulate at the surface. And depending on the ecosystem, we can have varying degrees of how thick this first organic layer is. And you can see that within the organic horizon, we have several sublayers, which consist of different stages of decomposition, partly decomposed and very decomposed. And that lower organic layer is something we hear of often called the humus Humus is a portion of the O horizon that actually is quite useful. We can use this type of material for composting, for mulch, for a variety of applications. The next layer here in yellow is the mineral soil, and that consists of quite a gradient from mineral soils that are mineral particles mixed with organic matter to almost no organic matter whatsoever. So in the A horizon, we have what we consider topsoil. This typically has a high commercial value because it's not only organic matter, but there's a significant mineral component, and therefore it has everything that a plant would need for growth. It has organic matter, it has nutrients. So topsoil tends to be what we're talking about when we're thinking about the growth of crops, the growth of plants, especially in a commercial application. We have this E horizon, which is leached or eluviated clays. So we're moving into finer and finer particles of mineral particles that are adsorbing to clay materials, very, very fine scale. In the B horizon, we have what would be considered subsoil. This tends to have a low commercial value. And below the B horizon, we have the C horizon, which is unweathered plant material. It has little to no weathering or accumulation. And by weathering here, we're talking about the physical breakdown of parent material or rock due to the force of water and other materials exerting a friction and frictive forces on the surface of this parent material and breaking off small quantities of that material into finer and finer pieces until we're really getting down to an atomic level. And underlying that C horizon, we have the regolith or the R horizon. This is bedrock. And this is really homogeneous stuff that's underlying the entire soil profile. So we can see a lot of different common soil profiles on our planet. And just for a few examples, we can take a look at tropical rainforests, temperate deciduous forests, and grasslands. And if we look at these soil profiles, we can see that visually they're very different. And the first observation we can have is from the O horizon. In tropical rainforest soils, we have a very thin or negligible O horizon because organic matter is decomposed and recycled very quickly. But when we look at temperate deciduous forests, we have some O horizons, we have some forest litter that transitions into a mixture of humus and mineral soils. And this is pretty consistent over time, and the presence of the O horizon is a characteristic of these temperate deciduous soils. So you might want to ask yourself, why do we have such a difference in the O horizon between tropical and temperate forest ecosystems? Think about that, and we'll get to it in a moment. But when we look at grassland soils, we see something very different. There is no O horizon because the organic material produced at the surface through grasslands and wildflowers, things of this nature, they quickly decompose into a rich humus, which tends to have a higher pH, very dark, rich, and fertile soils. So vast tracts of many continents are comprised of these types of soils, which have a good blend of organic and mineral soils, and this makes them very productive and very useful for agricultural activities. And if we look at the other layers, the other horizons in these soil profiles, we see that they are quite different depending on where we are, what type of biome we are in, what kind of ecosystem we are in. And in fact, the USDA, the U.S. Department of Agriculture, has characterized soils and soil structure all over the planet and been able to map the variation in soil structure in a very elegant way. So here in this map, you can see soil structure all over our planet, and you can see that it's incredibly heterogeneous. So that And we have many different types of soils that are all characterized, most of them characterized by a word that ends in SOLS or sol. And we can see that, for example, in some parts of the world, we have alpha sols, andisols, iridosols. These types of soils are, or these types of words are classifying soils using a taxonomic structure that's very similar to how we identify species of birds or fish or mammals, how we classify all of life. So the USDA has come up with a similar taxonomic framework, not for a living organism, but for the abiotic composition of soils. And taken as a whole, the soil structure anywhere on our planet is reflective of the precipitation and the temperature, which are both key factors in the rate at which parent material is weathered, and therefore dictates the structure of soils within any part of the globe. So global soil structure is really dictated by climate. How much rain do we have? How warm is it? How cold is it? And we can see this even more clearly if we compare these soil types with global biomes, different types of global ecosystems. So in the right-hand figure, we're looking at those same soils, but now they're organized in two dimensions across an axis of temperature on the x-axis and precipitation on, temperature on the y-axis, precipitation on the x-axis. So we can see that with increased weathering from the left to right, we change the types of soils. And due to that change, because of that change, in part, we affect the types of species, the types of ecosystems that can be supported by those soils. So on the left-hand side, you can see a graphic that's showing you the same axes, temperature as a function of precipitation, and these are including some of Whitaker's classic designations of terrestrial biomes. So if we line these things up, we can see that, say, Arctic tundra ecosystems, which are very cold and very dry, typically have gelosol-type soils. Gelosols are considered a type of cryosol, a cryosol meaning a frozen type of soil. And so these types of soils really have an impact on what species are present, what plant species in particular are present in Arctic tundra. At the other end, we have tropical forests, which are comprised of oxisols. These are heavily weathered soils that have very low organic matter content and a lot of oxidized minerals. So these types of soils underpin tropical forests, and they influence the type of plant species that we will find there. Most importantly, the rate of weathering and the temperature of an ecosystem is going to affect how much organic matter is present, because organic matter is subject to decomposition, and decomposition is strongly influenced by temperature. If anyone has any experience with organic composting, if you compost your food waste at home, or the waste from your gardening or forestry or anything like that, you know that you can actually create really rich soils by allowing plant material to decompose. In the process of decomposition, organic matter is respired by microorganisms, just like we would eat our dinner. They release CO2, they create water and generate some heat. And the further along they go in decomposing organic matter, the higher and higher the carbon to nitrogen ratio of that material becomes. So it influences its fertility, the soil's fertility, how much nutrient is available, and what types of plant species can utilize that soil for growth. And all of this decomposition is mediated by bacteria, by fungi, which are the classic decomposers, and even by some invertebrates like earthworms, which can feed on organic matter. So ultimately, the raw materials that we add include the organic matter, some minerals and nutrients and water, and we get the same kind of stuff uh, when we compost things, when we allow them to decompose. However, the biomass of microorganisms which will increase substantially, and the complexity of the material will decrease over time. Indeed, decomposition rate increases with temperature, and this is why I said earlier it's interesting that in tropical ecosystems we have a very thin or non-existent organic matter layer, or O horizon, whereas in temperate ecosystems we might have a substantial O horizon. The difference is due to decomposition. In the Arctic, we have almost no decomposition. In fact, there are vast tracts of land in Arctic ecosystems that are frozen in permafrost, so there's little to no decomposition whatsoever, and significant quantities of organic matter are present there. And one of the biggest risks identified by climate scientists is that as the planet warms and the permafrost thaws, it will become available for decomposition processes, which will dramatically increase the CO2 and methane emissions to our atmosphere, further complicating our global warming scenarios. And this is a positive feedback. So the more the planet warms, the more decomposition you have, and the more carbon compounds that will be released into the atmosphere. In the figure, you can see that decomposition transits through different phases. And you can see in these lines, this is the percentage of material remaining of these different components in the final product relative to the beginning. So at first, organic matter may be composed of a lot of cellular debris, so literally plant cells that are breaking down into their constituent parts. There are a lot of structural plant materials like cellulose and lignin. These are really complicated. They're not complicated, but they're very large carbon compounds that are difficult for many organisms to decompose. For example, if you eat a lot of plants in your diet, the cellulose and lignin are most likely non-digestible. This is what we would consider to be fiber in our diet, and which is considered good for our health, for keeping our guts clean. So those types of materials are difficult for all organisms to decompose, but some microorganisms can break them down slowly over time. And as they do process that organic material, the biomass of microbes will increase. So by the end of this decomposition process, a majority of the final product is derived from microbes, either their cellular debris, their organic compounds, their released nutrients present in that material. And I think the most interesting aspect of this figure is the x-axis. You can see that in the tropics, all of this can take place over the course of 3 years, whereas in the Arctic, it may take over 100 years for decomposition to proceed. So temperature definitely has a strong influence on the rate of organic matter decomposition. Now, how do we assess healthy soils, especially on a changing planet? And some of the key issues we're concerned about are soil fertility. How rich are the soils? How much productivity can they sustain? What types of plants can we grow in those soils? We're also concerned about erosion. How quickly is soil lost from the surface? Can we conserve it? Can we protect it from being washed away? And last but not least, contamination. How do we keep our soils clean and free of contaminants? And if they are contaminated, how can we utilize technologies and biology to remove those contaminants from our soils so that we're not transmitting those pollutants to ourselves by eating those plants? And especially the issue of fertility and contamination are driven by key soil property, which is called adsorption. Now, adsorption describes the phenomenon when a particle adheres to another particle. In this case, we're talking about cations, or positively charged ions, that are sticking to negatively charged soil particles. In this figure, you can see two types of soil particles. One is a humus particle, which is organic matter, decomposed organic matter, that has a slightly negative charge. And then we have a clay particle, so this is a mineral particle that also has a slightly negative charge, and it's surrounded by some soil solution. So one of the reasons that we water our plants is not only because the plants like to transpire water, but they also need an aqueous solution with which they can do some nutrient exchange or cation exchange with their surrounding soil particles. So for the soil, this adsorption is a physical process. It's simply the negative charge of these particles is attracting positively charged things. But for the plant, it's very much an active process. It's a selective process based on the plant's need for certain types of minerals in order to grow. So in this figure, you can see there are several plant nutrients that are quite important: magnesium and calcium, which can be used for reinforcing the structure of plant. We have potassium and ammonium, which are important plant nutrients that are vital for cellular chemistry and growth. And these types of ions are all adhering to our soil particles. Now, roots are an impressive structure created by plants. They can penetrate through different types of soils, but they're also influencing soil chemistry on very small scales. In this image, you can see that the root hair is surrounded by hydrogen ions, and so the plant root can actually pump these ions out into the soil solution. And these positively charged ions can overwhelm the negatively charged soil particles and even can bump or push some of the other heavier cations off of the soil particles so that they become suspended in the soil solution and available for the plant to take up and assimilate into its biomass. So roots are masterful at pushing cations around and making sure that the plant is getting the types of resources that it needs for growth and survival. Now, adsorption, the process of adsorption, is very influenced by the soil's pH. So under normal conditions, a negatively charged particle is going to adhere to all kinds of positively charged cations. But if we increase the amount of free hydrogen ions in our soil solution, we can push off all of the important macronutrients, and we can change the acidity of the soil itself, which can have positive and negative feedbacks into what types of ions are available. For example, if we look in this figure, we can see that some of the most important plant nutrients, like nitrogen and phosphorus at the top, they decrease in their availability to a plant as we increase the acidity of the soil solution. So as you move to the left towards the red, the availability of nitrogen and phosphorus to a plant decreases dramatically. And this is similar for many other plant macronutrients. But what's a big concern is that as we acidify soils by pumping carbon dioxide and sulfur dioxide in the atmosphere, which eventually makes it into our groundwater and changes our soil chemistry, we can also release certain types of cations from adhering to soils. So things like copper, zinc, aluminum, these types of metals can be released from soil particles. They can be assimilated by plants and consumed by animals. And this type of acidification can lead to these trace metals becoming toxic within the ecosystem and causing environmental problems and threatening human and animal health. So it's not good to have soil acidification because it's limiting a plant's ability to grow by limiting their nutrients, and it's also releasing toxic substances from soils that normally are bound to those soils. One key thing that we will work on in our class is measuring soil texture, because soil texture is critically important for the soil's ability to absorb nutrients and to retain nutrients within an ecosystem. It also has a big effect on what types of plants can grow within that soil. So here we're looking at the three major classifications of soils: sands, silts, and clays. You can see that sands range from 2 to 0.05 millimeters. Silt is 0.05 to 0.002 millimeters. And clays are everything that's less than 0.002 millimeters, so very, very small particles. The most important difference between these particles is their surface area to volume ratio. So sand, being a very large particle, it has a large volume, but relative to its volume, its surface area is very low. So it doesn't have, on a per volume basis, it doesn't have the same amount of negatively charged sites with which it can absorb lots of cations. So clays are the best at absorbing nutrients. They have the most charged sites for nutrients to adhere to. So they're really good for retaining nutrients. You can also see that the interstitial spaces, the space around these particles is different. So there's a lot of space around the sand, but there's very little space around the clay. And this space can be occupied by not only plant roots, which have to work harder to push through dense soils that are clay rich, but it also affects the transport of water through those particles. So because sand has a lot of space, water can move through very quickly. It can also take up a lot of those nutrients off of the sand surface and export them from the environment. So sand is really poor at retaining nutrients, not only because of its high surface area to volume ratio, but also to its susceptibility to leaching and having lots of water permeating the surroundings. So particle size affects the absorption of nutrients to soils. It affects the abilities for plants' roots to penetrate those soils, and it affects the ability of those soils to retain nutrients or prevent leaching of nutrients. And this week we're going to quantify soil texture in the laboratory. So on the right-hand side, you can see the tools that we're going to use. We're going to use a simple device called a hydrometer. Hydrometer is consider it like a measuring stick with a float on it. And this float provides some buoyancy in water that's going to change with the density of the solution. So if we add a soil to our water and mix it up very well, then the soil density is going to, if the soil density is very high, it's going to keep the hydrometer up higher in the water. And as the soil density changes, as particles begin to sink to the bottom, then the hydrometer will also sink because it's not being supported by those particles in solution. So we use the hydrometer to calculate the percent clay and the percent silt in our soil sample, and by difference we'll be able to calculate how much sand was in our samples. And using this soil texture triangle, which you can see on the left, we can classify our soils very simply based on the three parameters. And you'll notice that around the perimeter of the triangle, the soil types are names that consist of the dominant constituent. So if we have 100% silt and very little sand or clay, then we simply call that soil a silt. If we have anything below 40% silt and less than 70% sand, then that's going to be a clay. And clays tend to be a very large and common type of soil. And only when we have a nice mixture of all of those constituents do we have something different, something that we would call a loam. And a loam is a soil that is very well balanced, consisting of equal proportions of silt, clay, and sand. Now, the soil texture, its composition, particularly the abundance of clay in that soil, has a big impact on how other types of chemicals, other types of perhaps unnatural chemicals or man-made constituents are absorbed to soil particles. So here we can see a figure of organic carbon and pollutants that are absorbed in soils and how they can be exchanged with the environment. So certain types of organic pollutants can be taken up by plants, which can be used to detoxify the soil, or unfortunately those pollutants can end up in our crops and they can be removed and consumed by humans or animals. Sunlight can be important in removing pollutants by oxidation. We can have chemical decomposition or biological degradation that breaks down organic pollutants. We can have mineral reactions on mineral surfaces. We can have leaching into groundwater, et cetera, et cetera. So all of the things that we have talked about can influence not only plant nutrients, but also certain types of pollutants that may not have any function in the plant, but can be transported biologically to our dinner plate at some point if we're not aware of these complex processes. How does the soil become contaminated? Well, this is pretty easy. It's coming from urban and agricultural runoff, from mining, from industrial effluents, from atmospheric deposition, and from contaminated water. Basically, everything that we do careless in the environment can lead to the contamination and the pollution of our soils. Many of the case studies for soil pollution center around industrial applications, whether it's these could be like from drilling for oil on land, can lead to a lot of hydrocarbon contamination in surface soils. Natural gas extraction can contaminate groundwater. People in the U.S. are now concerned about fracking, which is looking for shale gas. This is a very intensive engineering of the surface of the planet, which is intimately perturbing the relationship between soils and hydrocarbons, and which can be toxic to life. Other sources of pollution from mining, for example, are very important, as well as industries that are using heavy metals like chrome plating or gun manufacturing, ammunition manufacturing. All of these things have a very poor history with preserving soil quality and increasing pollution. Now, we should mention erosion here, because erosion is a key and very important factor, especially with climate change and changing precipitation patterns. We have a real threat on our hands with soil conservation. In some parts of the world, soils are lost due to water erosion, sometimes lost to wind erosion. Sometimes it's a chemical or physical loss of soils. But almost all over the planet, with the exception of the high temperate zones and Arctic ecosystems, we have some sort of soil disturbance or loss of soil due to erosional processes. In the future, this will become increasingly a concern because, as I mentioned before, soil is a non-renewable resource. But we will not be measuring erosion in this week's activity. This week, we will be learning how to sample soil and prep it for some other research. We're going to revisit nutrient chemistry. We're going to try the phosphate analysis, but this time using soils. We'll also measure soil pH. And using the hydrometer, we will quantify soil texture, and we'll also show you how you can analyze samples for soil organic content analysis. So uploaded to the website, you'll find the lab protocol, which will detail some of these methods. We'll be demoing some of these sampling tools that we can use in the field to take soil samples and also separate those soils or prepare them for more downstream activities. So look forward to doing that. We'll also show you how to do a soil extraction where we're essentially hydrating soils with a solution that serves to push off all of the adsorbed nutrients on the soil particle, and then we can collect that extract and we can use it to measure things like phosphate concentrations. So you'll get a sheet like this that again takes you through the process of doing a phosphate an assay using a spectrophotometer. We'll hydrate soils and we'll measure the pH, the resulting pH, because the soil's cation content is going to help to change the pH of the water around it. So this is a very simple way of measuring soil pH. And soil pH has a key impact on plant health. Plants prefer pH that is around neutral, not too acidic, not too alkaline. We'll also demo how to use a muffle furnace for quantifying organic content of soils, and we'll let everyone have a chance to see this process firsthand and weigh some soils that have been muffled and had all of their organic material removed. So you'll get to see what is the difference between the organic contents of soils and their inorganic components. So that concludes the lecture for this week. Please make sure you download the protocol for the lab and review these materials. We have 2 video tutorials illustrating how to do some of the lab work next week. So please review those so you're familiar with them and ready to work on Tuesday. See you then.